glad to be here. So thrilled to be talking about this extremely important topic, and glad to see the curiosity from everyone here. The disclaimers, the important part: I am not a medical doctor, and this is not medical advice. All these views expressed are my own. They don't represent the views of my employer or any entity. And with that said, I am employed by Flagship Pioneering, which has a financial interest in Moderna, which is making one of the vaccines. And all information is public and non-confidential. So, this is going to give you insider knowledge on investments. I'm not paid to make any statements, and I'm not selling any products or services. And this is kind of an odd disclosure, but it seems to be important these days. This is not a presentation in favor or support of any political views. This is just the science. This is what we know. This is what we think. And this will hopefully help you make your own decisions. So a little bit about my background. I have an education in chemical engineering from UMass, then Northwestern, and then the University of Michigan. And it specifically, my PhD research was on modulating the immune response. So that's the adaptive and the innate immune responses, which are very important for vaccines. And so I will be citing scientific literature down here. And so you can look up some of these things if you want to read it for yourself. So the outline, I'm going to be talking about the significance of vaccines, the background of COVID-19, how it infects. So what is the process of immunity as it happens in your body? And how does it happen when we get a vaccination? And then really, I think a lot of people came here to learn about the vaccine types and look at the clinical trial data and pick that apart. And we won't be going into herd immunity, but if you have questions on it, we can definitely discuss that. But essentially the idea there is that we want to vaccinate enough people in our society so that we don't spread this virus uncontrolled. And then we'll have a Q&A session. Last time it was a ton of fun, so I'm looking forward to that. So the objective here is to provide you with scientific knowledge to empower you to scrutinize information about COVID-19 and COVID-19 vaccines. I'm not trying to proselytize. I'm not trying to tell you to go and get a vaccine. I'm trying to help you understand and make decisions for yourself. And really, the best thing you can do is just ask questions and not just absorb things you hear on the internet. And so that's my goal for today. So a historical perspective, if you look on the left side here, this is a hospital in Los Alamos. This is back in, I think, the 40s or 50s. And this is a row of iron lungs. And these were used to help people who were paralyzed by polio to breathe and sustain life. And polio is a terrible disease. And Salk discovered the polio vaccine. And this is the headline from 1955. And you see it just like the headlines today for BioNTech and Moderna, efficacy of 80 to 90%. And so what happened after that was all these polio cases and polio deaths just plummeted because there was national and global acceptance of this vaccine, and it was super effective. And so we were able to eliminate polio in the United States. And this is incredible. And we've almost been able to eradicate it from this earth. And there are some agencies like the Bill and Melinda Gates Foundation that are trying to do so. But still, misinformation about vaccines has hindered universal adoption. And so even though we've eliminated it in the United States, there's still cases in the United States, in the world today. So yeah, I want to draw a parallel. These machines, these iron lungs, which help people to survive by artificially helping them breathe, this is very similar to what we see in the ICU today, where people are hooked up to respirators, like terrible apparatuses or apparati that, I mean, it's really an uncomfortable situation to be in, and these people are fighting for their lives. So this is where we are today. We have these vaccines coming out, they're being authorized. The question is, what's it going to look like in the next decade or in the next year? Are we going to adopt these? Are we going to get effective use of the vaccine? So why do we even need one? A lot of people aren't convinced that COVID-19 is a deadly disease. They say, oh, it's just like the flu. I know someone who had it. They hardly even knew they had it. They were just diagnosed. It's not such a big deal. Well, this is the excess deaths report from the CDC. And so this starting in 2017, going up to today, these are the numbers of deaths in the United States. And this orange curve is the margin where we consider excess death. And so you notice the deaths are always or typically below that margin. And then last spring, deaths started to exceed that excess death threshold. So all these people who died would have been living today if it weren't for COVID-19. So that's really showing the significance. That's about 10% of the total deaths in the last year. We do need a vaccine because vaccines stop preventable death. And so there's been a tremendous effort nationwide and globally to create vaccines. And here I have 64 vaccines listed. This is from Cognitic, which used World Health Organization data. But I've seen as large as 85 vaccines in trials. And these are spread out across over 100 trials. And you see that this is an incredible endeavor, and we've made a lot of progress. And so we're going to go over those vaccines, but let's start off with a little bit of the fundamental biology of what this virus is and how a vaccine actually would work. So you've seen this picture, it's from the CDC. This is the coronavirus, and this is SARS-CoV-2. This is the virus, and it causes the disease COVID-19. The spike protein binds to receptors that line your airways, and it enters the cell with this privileged access. And so this virus has a membrane, and within that membrane is viral RNA. And so RNA is the information for making protein, and in this case, it's the information for making more virus. So viruses are cell hijacking replication machines. And so we're going to want to understand what is DNA, RNA, and protein. We're going to be talking about them throughout because there are actually vaccines that are DNA-based, RNA-based, and protein-based. But DNA is in every single one of our cells, and it's essentially a blueprint, right? It has all the information for all the cells in your body. If DNA is the blueprint, RNA is just the middleman between that blueprint and proteins. And so every cell has all the DNA, but say a cell in your pancreas, which produces lots of enzymes, it's going to be very different than a heart cell, which is more for mechanics and rhythm. And so each cell needs to know what genes to express, and that is in the RNA. So RNA is like a work order. If you're going to build a house, you get these work orders. All right, order the 2x4s. Okay, call up the framing team. They do the framing. Then you call up the plumber. They install the plumbing. These are specific sets of instructions, so you don't have to express every gene, but you can express specific genes that are important. So it's a temporary work order to produce proteins. And proteins are the biomolecules that do the work and provide structure in the body. So these would be the workers and all the materials. And so plants, humans, bacteria, they use all these things, DNA, RNA, and protein. They're all over the body. And RNA viruses, like coronavirus, just uses the RNA to create protein. And so here's how it hijacks cells for replication. We know that this is an airborne disease. Coronavirus, it enters the air from talking, breathing, sneezing, coughing, and these aerosolized particles get taken up through the airways, and the spike protein allows the virus to bind to the ACE2 receptor in healthy cells. And so then it enters the cell and it dumps out its RNA contents. So that RNA, remember, it's a work order to create protein. And so it hijacks the cell's protein machinery to make more of its own components. And so what you end up with is a cell that's now producing coronavirus in the body, and then it produces lots of it. It dumps it out, kills the cell, and those coronavirus particles then infect other healthy cells. And so it spreads. And so the immune system is going to detect that something's wrong, right? We have dying cells here. So we have these innate immune cells. They know when there's damage and they go and start cleaning it up. And they start secreting these molecules that attract other immune cells. So they end up secreting these signals, these danger signals, and that attracts other immune cells. And so here, this APC is an antigen presenting cell. And it's called that because it's actually going to take up the virus, digest it into little fragments, and present these fragments called antigens to other cells in the body. And so here comes the T cell. The T cells are the master choreographers of the immune response. There's many T cells. In fact, there's millions of them, and each of them recognized as a different antigen. So, every year you get a new flu shot. There's T cells that recognize the flu, but in this case, there's not going to be any interaction. They don't recognize coronavirus, so they just kind of float away. Then you have T cells that maybe recognize salmonella. They don't recognize coronavirus, so they just float away. But then you get this interaction. Finally, there's this T cell. It was born for this. It recognizes coronavirus protein fragments, spike protein fragments. And so there's going to be an interaction there. That T cell then gets excited, activated, and starts dividing, making more of itself. That T cell goes forth and goes and finds a B cell. We're talking about antibodies a lot in the news. That's talking about this receptor that is special to B cells. If that B cell has a receptor, an antibody, that recognizes coronavirus, then these T cells and B cells are going to get along and proliferate that response. And so what you get is B cells that start dumping anti-COVID-19 antibodies into the body. And so that's what protects you. They will secrete these antibodies in your blood for a long time. So you have a memory against this virus. And so the president had gotten infected with COVID-19. And when that happened, Regeneron was in the news quite a bit. Regeneron's technology was not a vaccine. It was simply a cocktail of these antibodies that bind to the virus specifically. And so you can kind of skip all this stuff and just inject those antibodies so that it binds to these, and it gives your immune system a little bit of a head start, a little bit more support. But it's not permanent, right? That Regeneron thing, it's a temporary solution. Your immune system didn't learn anything in the process. But this is quite taxing on the body to get sick like this. All these danger signals make you feel really sick. And what we found is not everyone's immune system works as efficiently as others. And so sometimes this process makes a mess of the body such that your lungs no longer work, you're inflamed, your temperature is high, and you actually will die without life support. And so there must be a better way to get immunity without risking your life. Instead of everyone catching the disease organically, like we're in the days of the Black Plague, we have vaccination. And so vaccines are a shortcut to learned immunity. So all vaccines have two components. One is an antigen, which is just a protein. And it's not just a protein, it is a protein that defines your virus. In this case, it's almost certainly the spike protein. And then you have this other component called an adjuvant or a danger signal, something that tells your body that's just not any protein, that is a very bad protein, and that we should attack it and we should learn from it. And so sometimes the vaccine has an adjuvant that is very explicit. It's a molecule that signals danger, or it's implicit. That is to say, your body just knows it's bad because it's being delivered in the form of a virus. And we'll talk about the details later. But essentially, in the vaccine, you have two components, the antigen, so the spike protein, and the danger signal. And just like we said earlier, cells take it up. They communicate to T cells, not any T cell, not the flu T cell, not the Salmonella T cell, specifically the coronavirus T cell. And we get proliferation, and they talk to B cells, and we get antibodies. And the immune system remembers. We have immune memory, and we have protection for a long time. Right now, we don't know how long is a long time. It could be a few months. It could be a few years. We currently don't know the answer to that, but we hope it's a long time. Here we skipped the cell death. We skipped the damage. We have a vaccine that essentially teaches your immune system what coronavirus looks like without going through all the effort and the life-threatening consequences. All right, so let's get into the types of vaccines we're talking about in the news and that are going to be unrolling as the months come around. So the first is RNA and DNA vaccines. And so these are the ones that are on the tip of everyone's tongue because these are the ones that are first to make it to the approval process. So we're talking about the Pfizer-BioNTech, which is approved, and the Moderna vaccine, which is likely to be approved by the end of the week. Those are both RNA vaccines. So RNA, we said, we have DNA, it makes RNA, RNA makes proteins. We are using this vaccine type to not deliver a protein, but deliver the RNA that will create the protein in the body. Because we said a vaccine has to have those two components, the antigen, the spike protein, and the immune activating signal. The beautiful thing here is that it was easy and quick to design, and that's why these two companies were first to the approval. The disadvantage is it's never been done before, and some people have been scrutinizing it because it's new and different. But then when we talk about going old school, these are the viruses or the vaccines that have been around for a long time. So that is the live attenuated and the inactivated viruses. And so these are actually both based on COVID-19. We're actually taking bona fide coronavirus particles and changing them. And so in the live attenuated, we essentially mess around with the RNA to make this a poorly tuned replicating machine so that it makes more mistakes and it's not as fit to survive in the body. But, and the good thing here is that it actually is quite robust because it's based on a real virus. But the disadvantages is that if you have a compromised immune system, this may not be the safest option for you. And so if you're immunocompromised, talk to your doctor about it. In the case of the inactivated vaccine, again, you start with bona fide coronavirus, but you kill it. You use radiation or chemicals to completely inactivate it and deliver that as a sign to your immune system, saying, look out for these proteins and attack it when you see it. And then you have the subunit vaccine. And so this is kind of like I described earlier, where you just deliver spike protein plus an adjuvant. And so some people are concerned with, what if the virus or the vaccine actually gives me coronavirus? In this case, it's most obvious that that's completely impossible. These spike particles will never be a virus, and you will never be able to get coronavirus from that vaccine. The final component is a viral vector. This is where you take some other virus that isn't a coronavirus, and you essentially put spike protein RNA inside of it. And that's where the Oxford, AstraZeneca, and Johnson & Johnson vaccines fall. And so when we kind of zoom out, what are the most popular designs and what are we going to be seeing in the springtime? It turns out most of them are protein subunit vaccines. So it's just spike protein plus an adjuvant. And then we have RNA vaccines, which I'm actually surprised there's so many of them, being an innovative technology. And so now we're going to do a deep dive and say, all right, who are the main players? What technology did they use? What are the clinical responses? Starting off with Moderna, they have developed an mRNA COVID-19 vaccine. This is the most zoomed out version of it. You essentially get an injection into your muscle of these lipid nanoparticles that contain mRNA. The nanoparticles then flow to your lymph nodes. They might passively flow there or get transported by an immune cell. But what happens in your lymph nodes is very important. So if you're ill and you go to the doctors, they often feel your lymph nodes, right? They're feeling for swelling and inflammation. Because when your body's fighting inflammation, they do it in the lymph nodes. This is where the cells divide, this is where they talk. And so these particles flow to the lymph nodes, they're taken up, and these cells actually produce spike protein. So remember, RNA is used in cells to create proteins. So what the beautiful thing about this technology is that it actually uses cells to produce protein on site in the body. And so these proteins that are expressed on site are going to be taken up by immune cells, presented to T cells, and a robust immune response will result. With this technology, vaccines can be rapidly designed and produced. So Moderna has been working on this technology for several years now. And so they have this platform already in place. So when you have an idea or a global pandemic just happens to come along, all you have to do is take this protein, whether it's a therapeutic protein or your spike protein, figure out what the amino acid sequence is, you throw that into a computer, and based on proprietary algorithms, they essentially do some RNA optimization. And they simply manufacture the vaccine by producing RNA and putting that in nanoparticles. It's as easy as that. Essentially, they're printing, almost digitally printing RNA code that's going to create a protein in the body. And so this is very exciting. RNA, it turns out, is extremely unstable though. So when this idea was conceived of a decade ago, people laughed like this couldn't be a medicine because RNA will never make it from the vial in the factory to the person it needs to help. And as soon as you inject it, it's going to be destroyed. It's completely unstable. We have RNA all over our body, and it's rapidly turned over. And it turns out a lot of the engineering was around creating stability in the RNA and making it stable in a nanoparticle. And so it turns out RNA is stabilized and delivered in lipid nanoparticles. And this is essentially like saying it's like a vinaigrette, really. You mix up oil droplets in water and they're quite stable. And I mean, that's exactly what it is. So your cell membranes are made up of phospholipids and cholesterol to stabilize. So are these nanoparticles. And then they throw in these ionizable lipids. So there's a fatty part and then a charged part, right? Turns out RNA is supercharged. And so this is essentially a mediator so that we can keep the RNA inside the particle. And then on the outside, you have this stuff called PEG. PEG is in so many things you don't even realize. This is what Miralax is essentially 100% PEG. What's cool about PEG? It loves water. That's why it works as a laxative, because it just absorbs water and passes right through your digestive system. And yeah, then we wrap up that RNA, which is engineered, and we put into the nanoparticles. We said RNA is super unstable. They've invested a lot into stabilizing it. But that's just the cool chemistry of it. This is the clinical data, which shows that it's safe and effective. And just a note here, this is not peer reviewed. I didn't read this in a journal. This is from their press releases. And also this is from the packet of paper that they shipped to the FDA for review this week. And so their phase 3 trial, it had over 30,000 participants. And those participants came from a diversity of people. So about 1/3 of people were from communities of color, and 7,000 were over the age of 65. And so this is extremely important. We're making a vaccine for the world, not just white males in America, right? We want to make sure that this drug works for everybody. And it seems obvious and it seems to go without saying, but it doesn't, because historically clinical trials had not been done in an equitable way. And it's something that the FDA is making a more concerted effort to put on their trial sponsors. But here's the cool part, the outcome. So people were injected with either a placebo, that is something that isn't the vaccine, something completely irrelevant, or the mRNA vaccine from Moderna. And what we find is 196 people ended up getting COVID-19 infections, and only 11 of those were in the RNA group. And so we're using this efficacy term in the news every day. Here's what it means. It's a relative efficacy, all right? And that means we're comparing people in one group to people in the placebo group, all right? So this is the vaccine group, 11 infections, placebo group, 185. So it's relative to each other, not percentage of total. And so it's 1, as in 100%, minus the relative infection rate. And so we end up getting, for Moderna, 94% effectiveness. And so you look at these two curves. The blue is the placebo. People are getting infected every day. The red, they have conferred this protective immunity. And so I look at this, I say, oh, it's clear as day. Why didn't the FDA approve this last week when we had all this data? Well, it's because they're really going to scrutinize this data for safety and efficacy. And it's not an easy decision, and they're not taking it lightly. So when you're concerned about your safety, is this safe? I personally think, all right, who are the people looking at it? What are the questions they're asking? They are asking every important question on behalf of the people. So efficacy is super high. It's consistent across age, race, ethnicity, and gender. And safety, it's well tolerated. There are adverse reactions, injection site pain, fatigued, myalgia, which is muscle soreness, arthralgia, that's joint soreness, headache, redness. There are side effects of these things. We'll talk about them later. It hasn't been approved yet, and the authorization will probably happen later this week, but that's for people over the age of 18, I think. And so they are starting a trial in adolescents. They already have started that. Pfizer and BioNTech paired up for this RNA vaccine, and it's reported to be greater than 90% effective. And so here's the phase 3 trial data. The trial had over 43,000 volunteers of diverse backgrounds listed here. Of the 94 cases of COVID-19, only 8 were in the vaccinated group. And so that's an efficacy rate of 95%. So there is some confusion here. There's 2 efficacy rates. One is a published peer-reviewed journal. But then as more infections came in the placebo group, that number has gone only better, and that's as of a week ago. And so when we look here, this is a 2-dose regime vaccine, and the placebo group is in blue. You zoom in, you can see between the 2 groups for the first about 12 days, there's no difference between those who are vaccinated and those who got the placebo. That's because there's a lot going on in the body over those first few days, right? The mRNA needs to produce protein. It needs to produce enough protein. That protein needs to be taken up by immune cells. The immune cells need to move. They need to talk to each other. They need to produce antibodies. All that takes time. So don't get your vaccine and just go out willy-nilly and say, I'm going to Disney World. These things take some time, and the effect only gets stronger after the second dose. And so you want to be vigilant, make sure you get that second dose. And so very good work from Pfizer and BioNTech. They have the only FDA emergency use authorization for a vaccine currently, and that's for people age 16 and plus. And so here's some advice for special groups, because these questions are bound to come up. I'm pregnant, I'm lactating. Can I and should I get the vaccine? No pregnant and lactating women were involved in the clinical trials, so we don't have data specifically for that group. The CDC's stance, though, is that these women should be given the option and choice to get a vaccine if they so choose, and you can look into that. People with allergies, so this is from the American College of Allergy and Asthma and Immunity. It's very rare, to have an allergic reaction to a vaccine, about 1 in a million. But it's not impossible, of course. Individuals with common allergies, to medications, food, inhalants, insects, latex, they say they're no more likely than the general public to have an allergic reaction to the Pfizer-BioNTech vaccine. However, they say you should be administered in a healthcare setting where you could actually manage symptoms of anaphylaxis, and you shouldn't get it if you're allergic to PEG. So some people are allergic to PEG because it's in so many products, and in that case, you should probably not get this. And yeah, that said, there were a couple of reactions in the clinical trials. Oh, those people were hypersensitive. So if you're a hypersensitive person, you're allergic to many things, they say just proceed with caution. So those younger than 16, there hasn't been a vaccine authorized for that. There are studies ongoing for ages 12 through 15. Okay, moving on to vector-based vaccines. So this is interesting. So when I say vector-based, we're talking virus-based vaccines. And so Codagenics is developing a live attenuated vaccine. And this was really interesting. I wasn't aware of this until I read about it. It's an intranasal, which is nice. The route of administration is favorable. It's a single dose, which is great for compliance. And this was super interesting. Here, they've taken the coronavirus, the bona fide virus, and what they've done is changed the RNA sequence in such a way that the amino acid sequence hasn't changed. That is to say, it codes for the same proteins, but by changing the sequence of the RNA, they've de-optimized how well the proteins can be translated. And so, to put it simply, they've done this really clever thing where all the proteins are the same as the coronavirus, but they did it in such a convoluted way that it's so inefficient. It's like a poorly tuned machine. This virus will essentially not reproduce well enough in the body, and your immune system will defeat it. So really cool, really interesting. It's pretty far behind though. It's only in phase 1 trials. And so they have a ways to go, but very interesting. Like I said earlier, if you're immunocompromised, this isn't the vaccine for you because it is based closely on the actual virus. Sinovac out of China is developing a chemically inactivated viral vaccine. And again, we start off with the bona fide coronavirus, and then you treat it with a molecule called beta-propiolactone. And so I, despite being a chemical engineer, don't know much about chemistry, but when I see this molecule, I know it's quite reactive. It is one angsty molecule. It has these bonds which are quite constrained. You don't see bonds shaped like squares in nature, and then the oxygens and the double bonds. You just don't see it very often. It's highly reactive, and it's a carcinogen, which, you know, when I first read this concerned me, but it's a carcinogen because it's going to chemically react within the virus particle and the proteins on the coating. And so this is going to completely chemically inactivate the virus. So essentially it's a dead virus when they give it to you. But because it's dead, it's not going to by itself elicit a strong response. And so they adjuvant it by adding aluminum hydroxide. And so this is a classic adjuvant. It's used very often. So you get 2 doses, antibodies are generated, but we won't get our efficacy data until January at the earliest. And then here's the evidence that beta-propiolactone actually does deactivate. As you increase the concentration of that chemical, you completely deactivate virus. That's not in coronavirus though, that's in H3N2. And then AstraZeneca has teamed up with Oxford University to develop a replication deficient adenovirus vaccine. So that's a very verbose way of saying it's not a coronavirus, it's a different type of virus. And what it does is deliver DNA for expression of spike protein within your cells. And so it's replication deficient, so it's not going to create more virus in the body like a coronavirus would, but it will produce that spike protein. And because a cell doesn't like being taken over to produce protein, it naturally adjuvants itself. So we consider it immunogenic, it stimulates immunity, which is a good thing in the case of a vaccine, but others want to use adenoviruses for other things. And it's challenging to inject these more than once because your body, once it sees it once, it learns from that, it doesn't want to see it again. Okay, so the AstraZeneca Oxford safety and efficacy looks promising. Here we have the placebo is actually a different vaccine for meningitis protein. Here, the vaccine is actually called CHAD. So CH comes from chimpanzee. So this adenovirus comes from a chimpanzee virus. They pulled out all the guts that make it harmful. They threw in the therapeutic version of spike protein in there. And what we find is it's looking pretty effective. And so there's two different scenarios here. The first scenario is they gave them a low dose, then they gave them a standard dose. And then the second scenario is 2 injections separated by time of just standard dose. And what we find is in the low dose, then the standard dose, we have 90% effectiveness. In the 2 times standard dose, you get 62%. So this had a lot of people scratching their heads. Why does the low dose and the standard dose have greater efficacy than 2 standard doses? We don't know. And the thing that was also getting us to scratch our heads was, why was it designed this way? And it turns out it wasn't. So it was supposed to be 2 standard doses, but the contract research organization that AstraZeneca had hired, made an error and accidentally gave a low dose. This seems fortuitous. Okay, wow, now we've discovered a higher effectiveness rate. But the FDA doesn't like inconsistencies, and they don't like you conducting a trial and then making a conclusion after the fact. It's called post hoc analysis. If anything, you want everything to go smooth as butter. You don't want any inconsistencies, and so they have to figure this out. This 90%, it could be true that a low dose and a standard dose has a better efficacy. We're gonna have to do more research to figure that out. It also could be because these numbers are so low at the moment. All right, Johnson & Johnson, and also known as Janssen, is producing an adenovirus vaccine. First of all, they have a 1-dose regime in clinical trials, so that's exciting. You only have to go in once to get 1 injection instead of 2. And then the other thing that's interesting is their clinical trial originally had 60,000 participants planned, but because the infection rate in the United States is just so rapid and so high at the moment, they can get statistical information by reducing their enrollment population by 33%. So it's kind of sad that that's the case, but it's also means fewer resources have to go into that clinical trial to draw the same conclusions. And then finally, subunit vaccines. So Novavax is the one getting most of the press about their subunit vaccines. Again, this is you deliver an antigen, which is the spike protein, in an adjuvant, which is a chemical or entity that stimulates the immune system. And so in this case, they use an insect cell line to cleverly create this protein, the spike protein. And the clever part is they get them to all stick to each other like this. So you end up with a spiky little nanoparticle of protein. So that delivers a lot of your antigen in a small little package. And then the other interesting bit here is the adjuvant is made of saponin, which is derived from a Chilean soapbark tree. And we've all probably consumed some of this at some point in our lives. This is what makes root beer foamy, and Slurpees, it gives Slurpee texture for those who love Slurpees. I know there's one in the audience. And so, yeah, they actually had to, demand overnight just skyrocketed for this pretty rare compound. The nice thing about the subunit vaccine is the storage, only 2 to 4 degrees C. So that's refrigerator temperature. And so far it's well tolerated, produces antibodies in phase 1 and 2 trials. So yeah, to summarize, vaccines need 2 things: the antigen, spike protein, and a danger signal. And there's several ways you can do that, right? One is by delivering simply the spike protein in an adjuvant, or you can deliver that spike protein in just a dead virus or a live attenuated virus. Or you could do something clever and innovative, like deliver DNA or RNA, which will create the protein in the body. The important thing here is that you're teaching the immune system to recognize spike protein so that when you encounter COVID-19, it's recognizing it, not for the first time, but already well prepared to fight it. We're transitioning into the things that are on everyone's minds, and then we'll get right to your questions. So important, super important, side effects are expected, and they are a result of the vaccine doing its work. So hundreds of thousands of injections have already been made across the breadth of these vaccines. No serious health problems have been encountered. There have been many side effects though, okay? And these side effects include redness, soreness, headaches, chills, muscle pain. These side effects of the vaccine can actually resemble COVID-19 symptoms, right? The symptom list for COVID-19 very much resembles the side effect list for vaccines. They're both using your immune system, right? So your vaccine has to stimulate immunity, and it's using immune cells and signaling molecules to do so. So you might feel a little bit lousy for a day, and this is completely natural. That is your immune system mounting a response to it. However, this is a walk in the park compared to getting COVID-19. These side effects are small in comparison to actually contracting the disease. But of course, if you have an emergency after getting a vaccine, seek out medical care. Call 911 if you have to. I would say the vaccine is much less harm than the actual disease. And I want to encourage everyone over the next several months, scrutinize unconfirmed reports of adverse events, all right? Someone tells you something and they say, yeah, I saw it on internet, YouTube, Facebook, Instagram, Twitter. You really got to dig into what is the credibility of this resource, who's producing this, why should I trust it? So please do scrutinize unconfirmed reports. Like I said earlier, there was seems like an allergic reaction to one of the BioNTech vaccines today in Alaska. Let's keep an eye on that. Let's find out why. Let's find out if it was related and how it might apply to our lives. Who gets the vaccine first and why? So the CDC has given guidelines recently that healthcare workers, so there's 21 million of them, should be the first priority group, and as well as people from nursing homes and long-term care facilities, which is about 3 million people. Healthcare workers, it's kind of obvious. If you're deadly sick with coronavirus, then you funnel yourself and you funnel this population to healthcare centers. So these healthcare workers are essential for rehabilitating these people, and also they're at risk because we're concentrating the virus in their work environment. So we want to protect them, obviously. Nursing homes and long-term care facilities, these people are at risk because they're old, which means their immune system is less efficient than young people. And they account for about 40% of COVID-19 deaths. So they have a tremendous need. So that's where the CDC guideline stops. The rest, it's up to the states to essentially decide how they want to order it. And the CDC has kind of hinted that essential workers should be next in line. So that's almost 90 million people, people with comorbidities, people over 65, et cetera. And so this is based on ethical principles from the CDC. Maximize benefit, minimize harm. So when we look at by age group, essentially decade by decade, and all the deaths, people 85 plus, I mean, they account for most of the deaths, and that deadliness is proportional to age. So we really need to protect the most vulnerable, which is older people. This is just remarkable. Percentage of COVID-19 deaths, out of all deaths this year, COVID-19 accounts for 6 to 10% of all deaths amongst all groups. So this is just staggering. I mean, this is a tremendous problem, and the vaccine is designed to change that. I was writing out the title to this. I almost wrote incredible vaccine development timeline, but it has been so credible, right? So they have done it at an astonishing pace, while maintaining the high standards we have for safety and efficacy. And so where a vaccine might take over a decade to develop under normal circumstances, we've been able to cram this into less than a year. And so this was prepared by the New York Times back in the springtime, and it's been incredibly accurate. And so what this took was innovation, so new technologies, and getting creative with resources, prioritizing important factors, and then regulatory efficiency. The FDA did not lower its standards. It has just had to be a lot more efficient. And so we have clinical trials, instead of waiting for one to finish before starting another one, we've been staggering and overlapping. And this is about accurate. By August 2021, that's when perhaps young people who are healthy will be getting their vaccines. And the approvals are starting to trickle in right now at the end of 2020. The summary is that vaccination is a shortcut to learned immunity, and it has the potential to save many lives. Thank you all for sticking around. I know it's a long-winded presentation, try to cram it all in. I'd love to take some questions if you have any.